Hello, Aniket. Only one person has joined, that is fine. So, I'll introduce myself. So, my name is Chaitra Tolankar. I am a PhD student. I am a fourth year PhD student. Okay, and I am from IIT Bombay, from Chemical Engineering Department. And I studied biochemistry and microbiology. That is my background, master's and bachelor's background. So, can you guys introduce or tell me what is your background? Or you can type also if you want. Please let me know about your background. You can unmute yourself and okay, MSc Life Sciences, and the other person, Della Abraham. So, please let me know your background so that it will be easier for me to explain um, the concepts. BSMS Biology. Okay, nice. So I'll again introduce myself. I am Chaitra Todankar and I'm doing PhD in IIT Bombay Chemical Engineering Department, and I am from microbiology bachelor's and master's in biochemistry, and now I am working on systems biology. Okay, so Madan ji, so what is your background, may I know? You can type in chat also if you want. I just wanted to know what are the background of students are here in the meeting. MSc Biochemistry. Okay, good. So even I did my masters in biochemistry from MS University Baroda. Um, so there are few students who are joining. We'll wait for 2-3 minutes and then we'll start. So let's start with the today's session. So the course name is Enzyme Sciences and Technology, and this course is taken by Professor Vishal Trivedi from IIT Guwahati. Okay, and I am the NPTEL TA for this course along with one more TA. And my name is Chaitra Todankar and I'm from IIT Bombay Chemical Engineering Department. Okay, so let's begin. So what are enzymes? Enzymes are biocatalysts. Okay. So enzymes are biocatalyst, and life processes or any reactions, all the reactions in the cell are carried out by enzymes. So all the reactions which occur in the cell are carried out by enzyme. So I'll give you a brief introduction about of all the 5 lectures which have been taught in the previous week, okay, and then we'll proceed on to the assignment questions. So and all the reactions inside the cells are enzyme catalyzed, okay, are enzyme catalyzed, and all these reactions are enzyme catalyzed, and enzymes are nothing but they are biological catalyst, okay, biological catalyst, and they are mostly protein in nature. So and these all reactions include metabolic reactions. They include metabolic reaction. They also include different types of molecular biology reactions like replications, transcription processes, translation, all the biological processes and physiological processes. These all are catalyzed by enzymes. Okay. So enzymes are biological catalysts, as I mentioned, and they are essential for all the life processes, right? All the life processes like they are essential for all the life processes, which includes replication, metabolic reactions, transcription processes, translation processes. All these life processes which are essential for the cell survival are carried out by these enzymes. Okay. Now there was and the study of enzyme is known as enzymology. Enzymology is study of enzyme, okay, and there are different so this enzymology was evolved around many years, okay. For past 100 or so years this enzymology field has been evolving. So first Splazzani was the scientist. He observed cell-free activity, okay. So he observed that that without any presence of cell, okay, there was a activity observed, okay, and this activity was just like catalysis, okay. At this time there was no catalysis term coined. So next in 1812 Kirchhoff observed that proteinaceous components, so the protein component of wheat was capable to convert starch into sugar, okay. So we have starch, which is a macromolecule, and it is converted into sugar, that is glucose, okay. And this conversion cannot happen just cannot happen in a test tube. We need some catalyst, okay. So there are biological catalysts, and this was observed without any presence of cell. And later there was a Swedish scientist, okay. He coined the term catalysis, okay. This catalysis term was coined by the Swedish scientist. Then Lusch described the salivary ptyalin enzyme. So this ptyalin enzyme is responsible for the digestion, okay, and he described the diastatic action of this salivary ptyalin. So this salivary ptyalin enzyme is found in the saliva, and it is capable of digesting, okay, it is capable of digesting the food which we consume, okay. It is capable of digesting the food which is consumed. So it will digest starch, protein, or other mostly starch, and it will convert into sugar, okay. And the first enzyme which was discovered was diastase, okay. So what they found out, that diastase they found out this enzyme from the bile juices. So they treated this when they found out that this bile juice contains this enzyme diastase, and this enzyme is capable of digesting different macromolecules like starch, protein, and lipids. Okay, these are the major macromolecules. Okay, starch or carbohydrate, we can say carbohydrate. Okay, carbohydrate, proteins, and lipids. So this diastase protein was found from the bile juices and from animals, and this enzyme was able to digest the different macromolecules. Okay, which are these are the three different macromolecules: carbohydrate, proteins, and lipid. Next, there were further discoveries in enzymology, which includes lock and key model. Okay, in order to explain what is the enzymatic mechanism or what is the reaction mechanism. So there were many discoveries like lock and key model and configuration model, and there were other discoveries like cell-free fermentation, cofactors discovery, induced feed model, cooperativity, etc. So why do we need to study this enzymology? So enzymes are very important part of the biological processes. Also, they have a huge application in industries. Okay, so that's why any enzyme which is very efficient in catalysis is like a gold for industry. So it is very important to study these enzyme properties, and how can we make better and better enzymes for greater production. Okay, in industrial application. Also, these enzymes are as a major part of different biological processes. They also control these biological processes, and thus these are very important to study. Okay, now the next question is that how these enzymes work. Okay, so this reaction is the reaction coordinate and how this reaction proceeds. Okay, so we have on the y-axis we have free energy. Okay, G, this G is the free energy. Okay, and this delta G plus plus is not free energy, and we should not confuse this with the free energy. This delta G plus plus is nothing but activation energy. Okay, it is activation energy, and it is not free energy. Okay, so what is this activation energy? We have reactant A. So the reaction here is we have reactant A, and it is proceeding to give us product which is B. Okay, the product is B. So now the reaction proceeds. This is the reaction coordinate. As the reaction proceeds, it forms a barrier. Okay, this barrier here, this peak is known as the transition state. Okay, the peak of this barrier is known as transition state. Okay, transition state. Okay, and that at this peak or the energy hill at this point, okay, the it can decay into either reactants, that is it can go backward, or it can go forward into formation of products which is B. Okay, so this is very important critical barrier or critical point, transition state, so that either there is possibility that there will be formation of reactants or there will be formation of products. Okay, now the blue line here, okay, this blue line here, it represents the uncatalyzed reaction. Okay, this blue line represents uncatalyzed reaction, and the red line here represents catalyzed reaction. Okay, this red line here, red reaction coordinate graph represents catalyzed reaction. Okay, so what is the difference between this catalyzed and uncatalyzed reaction here? The activation barrier. Okay, so this is the activation barrier of catalyzed reactions. Okay, this is activation barrier, that is activation energy difference of catalyzed reaction, whereas this is the activation energy difference of the uncatalyzed reaction. So as we can see from the graph, the difference in the activation energy of catalyzed is very less as compared to that of the difference in the activation energy of uncatalyzed, right? So what are the catalysts or what are the enzymes doing? They are reducing. So enzymes or any catalyst, what they are doing, they are reducing the activation energy barrier. They are reducing activation energy barrier. Okay, they are reducing this activation energy barrier. Okay, so they are not affecting the equilibrium of the reaction. Okay, we have a equilibrium here. They are not affecting this equilibrium, that is transition state, we can say roughly. They are not affecting the transition state, but they are lowering the activation energy. They are reducing or lowering the activation energy barrier. Okay, very simple. So thus it is lowering the activation energy barrier. The enzymes are very efficient catalyst. Okay, they catalyze reaction faster, right? And also they catalyze reaction faster, and they are very specific also. Okay, next, what are the enzyme properties? So enzymes are protein in nature except ribozymes. So ribozyme is nothing but RNA. Okay, any questions so far from the previous slide? Any questions? Please feel free to like stop me in between and unmute yourself and ask me the questions. Okay, so I won't be able to see the chat box, so you have to unmute yourself and ask me the question. Okay, any questions? You can respond if you want. Okay, if no more questions, then we'll move forward. So so enzymes are protein in nature except ribozyme. So ribozymes are have RNA. Ribozymes are nothing but RNA. Okay, the major macromolecule here is the nucleic acid, that is RNA. Okay, apart from this ribozymes, we have DNA enzymes also. Okay, so there are DNA enzymes also. You can search for different DNA enzymes. So RNA and both DNA can act as enzymes, and most of the enzymes, almost 90-95% enzymes are mostly they are protein in nature. Okay, so this is the structure of enzyme. This is a protein enzyme. Okay, and this is the red region here. This is the active site, and we have a substrate molecule as shown here. This is the substrate molecule. Okay, this is the substrate molecule, and this is the active site of the enzyme. So the substrate binds to this specific pocket of active site. It is made up of few amino acid, around 4 to 5 amino acid. It's a pocket where this substrate can come and bind. And enzymes are very specific towards the substrate. Okay, and now this specificity can be because of three different properties. Okay, it could be because of geometric specificity. What is this geometric specificity? It means that suppose the enzyme pocket is kind of like a curved pocket, so that a circular or a curved substrate will go and bind into this specific geometric pocket. It will go and bind to this specific geometric active site. Okay, so this is the geometric complementarity. Okay, and then we have electronic complementarity. What is electronic complementarity? That is, in the active site, as I said, that could be 4 to 5 different amino acids, and these 4 to 5 different amino acids may have different electronic charge on them. They could be either positively charged or they could be either negatively charged. Okay, they could be either positively charged and they could be either negatively charged. So for the substrate to come and bind, suppose in the active site, if the active site has a positively charged amino acid, okay, if it has a positively charged amino acid, the substrate should be a the substrate should have a negative charge on it, okay, so that there will be a force of attraction between them, okay. If both are positive, they will repel each other, okay, right? If they both are positive interactions or positive groups on them, they will repel each other and there won't be any interaction among these enzyme and substrate. But if the active site has positive amino acid and the substrate is negatively charged, there will be a electronic complementarity. And then we have stereospecificity. So what is this stereospecificity? That it is the spatial specificity which is observed in the enzymes. So mostly enzymes are protein, and protein is made up of all the protein is made up of amino acids, right? And these amino acids are L form of amino acid. So what is this L form of amino acid? So we have a amino acid which is a chiral, it has a amino group, it has a hydrogen here, and it has a carboxyl group, okay. That's why we call it as a amino acid, because it has a amino group and it has a carboxyl group. So the L form of amino acid refers to the this hydrogen. It's how the spatial position of this hydrogen is present in the, or this four different molecules are oriented in the space, okay. So there are L form of amino acid and there are D form of amino acids also, okay. But in nature or in humans, we have only L form of amino acids, okay. All the animals have L form of amino acid. D form of amino acid is only found in bacteria, okay. D form of amino acid is only found in bacteria. So bacteria has both L and D form, but all the mammals and vertebrates, they will have L form of amino acids, okay. Now catalysis rates are much higher. So catalysis occur at very faster rates, okay. The rates are in the range of 10 to power 6 to 10 to power 12, okay. These are the rates. So if suppose the uncatalyzed reaction is occurring at 1, the speed of reaction is at 1, so so the catalyzed reaction is 10 to power 6 times or 10 to power 12 times faster than the uncatalyzed reactions, okay. So they are very fast catalyzing reactions, and all the enzymes, they work under optimal temperature and pH, okay. This optimal temperature and pH is required for the reaction, and this optimal temperature and pH could be a range at which they can work. So suppose this enzyme pepsin and trypsin, chymotrypsin, they are present in our stomach to digest different proteins, okay. Trypsin enzyme is present in the stomach, or chymotrypsin, it is present in the stomach to digest the different types of protein, and the pH in the stomach is around 0 to 1 pH, okay. So the optimal pH for the trypsin enzyme is 0 to 1, whereas the enzymes which occur in the physiological in the cell at physiological pH, that is 7.2, mostly all the enzymes have this optimal temperature, that is optimal pH, that is 7.2. Exceptions are there like trypsin, chymotrypsin and pepsin, which are found in the acidic environment like stomach, and the optimal pH for them is 0 to 1, okay. So similarly we have different optimal temperature, but most of the enzymes will work good at 37 degrees Celsius, okay. So whenever we get fever, the temperature goes up to 40 degrees Celsius, that is approximately 104 degree Fahrenheit, okay. That is considered as high fever. So at that time these proteins can dysregulate, okay. That's why we say that, that's why we reduce the temperature, okay, whenever we have fever, so that there is proper functioning of the body, right. And enzymes can also be modified okay, by allostery. So what is allostery? We'll learn this in the future classes, what is allostery. But enzymes could be modified, they could be either phosphorylated, methylated, acetylated, okay. There could be different covalent and non-covalent modifications on the enzyme, okay. One of this is allostery, okay. Allostery could be either positive or negative allostery. So positive allostery will increase the binding or specificity or the reaction rate, whereas negative allostery will decrease the rate of reaction of enzymes, okay. Any questions? Okay, then so enzymes require different cofactors and coenzymes in order for them to function. So cofactors are generally ions, okay. They are inorganic ions. Cofactors are inorganic ions like copper, Fe2, Fe3 plus, potassium, magnesium, okay. So remember that cofactors are mostly ions, okay. And these are the different ions. For example, Cu2 plus is found in cytochrome oxidase, okay. This cytochrome oxidase is found in electron transport chain, okay. Similarly, we have Fe, Fe2 plus, Fe3 plus, okay. So these are the inorganic ions are the cofactors, okay. And then we have coenzymes. So coenzymes are generally vitamins, okay. Coenzymes are generally vitamins. They are the organic part, okay. They are the organic part of the enzyme. So cofactor and coenzyme together work, okay, in order for enzyme catalysis. So the coenzyme here, for example, is coenzyme A, okay. So coenzyme A will transfer acyl groups, okay. It will transfer acyl groups, and it is derived from pantothenic acid, okay. Similarly, we have coenzymes derived from vitamin B12 and vitamin B2, okay. So mostly all the coenzymes are vitamins in nature, okay. They are derived from different vitamins, okay, or they could be some vitamins, okay. So here directly vitamin B12 is used in the alkyl or hydrogen atom transfer in different reactions, okay. Then how do we classify this enzymes? So enzymes are classified into these 6 different classes, okay. This is very important. And how did we decide this 6 different classes? These are decided based upon what type of reactions they catalyze, okay. Different type of reaction they are catalyzing. So the numbering here is important, okay. The numbering here is important, and this classification is known as EC classification, also enzyme classification, okay. So this is the EC number, EC1, EC2, EC3, and so on. So this order is very important, okay. This order is set by the all the biochemist and the enzymologist, okay. So this order is important for enzyme classification. So the class 1, that is EC1, is oxidoreductase class. So what type of reaction do they catalyze? They catalyze transfer of electrons, or they could also transfer hydride ions or hydrogen atoms, okay. The first is very easy. They carry out oxidation and reduction reactions, okay. So oxidation could be they can carry out oxidation or reduction by either transfer of electrons, or they can reduce a substrate by adding hydrogen atoms or removing the hydrogen atoms, okay. They can either reduce or oxidize the substrate depending upon what the reaction the enzyme is catalyzing, okay. And the next reaction, next EC2, is the transferase, okay. What are these transferases? So as the name suggests, transferases, they will transfer group, okay. They will transfer this different functional groups. Okay, so this functional group can be either phosphate groups, okay, it could be either methyl group, okay, it will transfer different functional group like amine group. So these are the different functional group that these class of enzymes will transfer onto the substrate. That's why the name transferases. Okay. Then the next class of enzyme EC3 are hydrolases. So as the name again, hydrolysis reaction, it will carry out hydrolysis reaction. These class of enzyme hydrolases, they will carry out hydrolysis reaction. So what are the hydrolysis reactions? So these reactions are basically breakdown reactions. Okay, these are the breakdown reaction. That means it will break the two substrate. Suppose I have A and B together. Okay, I have A and B together. A water molecule will be added in between. Okay, there will be a water breakdown of these two substrate. Okay, and a water molecule will be used. Okay, to get products, to get the product A plus B. Okay, this type of reactions are breakdown reactions. Okay, and this breakdown will be with the help of water molecule. Okay, the breakdown will be with the help of water molecule. This water will be added in this within these two covalent bond, and they will be broken down into two different products, which is A and B. Okay, so these are the hydrolases class. Then we have lyases. So lyases also is a breakdown, is also the enzymes which break down different molecules. Okay, what they will do, they will cleave CC bond, CO bond, CN bond. Okay, they will cleave these bonds. Okay, and so here the water molecule is not involved. Okay, so there is cleaving of these these different bonds in absence of, okay, this is very important, in absence of water molecule. Okay, so the only difference between hydrolases and lyases is in hydrolases we use water, whereas in lyases we don't use water. Okay, in absence of water, there will be cleavage of CC bond or CO bond. Okay, then we have fifth class of enzymes, isomerases. Okay, what are these isomerases? It refers to intramolecular rearrangement or isomerization, okay, of different molecules. So suppose I have molecule A, B, C. So the isomerase will it will not change the molecular weight. It will just rearrange these molecules into CAB. Okay, so there is just rearrangement or intramolecular rearrangement. All these types of reactions are catalyzed by isomerases. They can also catalyze cis-trans isomerization, or mutases can rearrange the different functional groups on the either side of the carbon atom or any other molecule. Okay, then we have ligases. So ligases is only one enzyme which forms instead of breakdown, it leads to formation. Okay, it's the class of enzyme which leads to the formation of different macromolecules. Okay, so they are just opposite of lyases. Okay, so lyases is breakdown, whereas ligases is formation of macromolecules or molecules, and they will form CC bond, CS, CO, CN bonds. Okay, by condensation. Okay, and since it is formation of some molecule, they will require ATP. Okay, they will require, so any formation of molecule, any formation of bond will require energy. Any formation of bond will require energy, and this energy will be provided by the ATP. Okay, or it could be any cofactor. Okay, any cofactor or ATP is required. So any energy in form of ATP or other cofactor like NAD, NADPH, okay, it is required for the catalysis of the ligases. Then we have enzyme classification number. So here this EC number. Okay, so we have the enzyme can be classified into 4-digit EC number, for example 1111, and this 111 has its own significance. And what are those? So the first one here, what it means what EC class it belongs to, whether it belongs to oxireductase, whether it belongs to transferase, whether it belongs to hydrolases or lyases or isomerases or ligases. Okay, these are the different classes of enzyme which we studied in the previous slide, different EC numbers. So if the first number is 1, it will belong to the class oxireductase. That means it will carry out either oxidation, reduction, or both. Okay, both redox reactions. So here it belongs to the first number will tell it will belong to which class. The second number here refers to which subclass it will belong to, and this subclass, it means for example in oxireductase, let's take the example of oxireductase. The subclass in oxireductase is which functional group, like it will which functional group it will catalyze the reaction with. So in the oxireductase, it will catalyze the functional group of alcohol. It will only reduce or oxidize the alcohol molecule. Okay, so this subclass B, it will tell us about the substrate information. Okay, the subclass here, it is telling us about something about the substrate, that at what the enzyme will act on which substrate. Okay, so it will act on the alcohol group. Okay, it will act on the alcohol or OH groups, CH2OH group. It will act on the alcohols. The third number here refers to the subclass, and it refers to what will be the electron or the electron carrier or hydrogen ion transfer. So here the hydrogen ion transfer, it could be anything. It could be NADP, NADH, or it could be some coenzyme. Okay, so this will tell us about the subclass will tell us about who is the electron carrier, or are there any cofactors or coenzymes involved. Okay, and the D here, it refers to the what type of reaction it is. It will give us more information about what type of reaction and then where it is involved in. Okay, so let's take this oxireductase example. Here the oxireductase, the EC number is 111. So 1 refers to oxireductase enzyme. Okay, it belongs to oxireductase class. Okay, the second one, it will tell us about the substrate. So the substrate here is involved is the alcohol group, as I mentioned, so which is CH2OH. That means it will reduce or it will oxidize this CH2OH group. The third one is what electron group or electron transfers. Okay, which is what are the electron transfers. So the electron donor here is NAD or NADP. Okay, NAD plus or NADP plus. So if the electron donor is either NAD or NADP plus, then it will have a number 1 here, and the electron donor could be either FAD also. Okay, sometimes it could be another cofactors like some cytochrome oxidases or different quinones. Okay, it could be other quinones also. So there could be different electron donors, but here if it is NAD plus, it will have the value of 1. And the third here, 1 refers to the subclass, that is what it is doing. It is doing alcohol dehydrogenase. So the reaction is dehydrogenase, that is it is removing the hydrogen. So the reaction is alcohol dehydrogenase. That is, the alcohol group is converted, this CH2OH group is converted to aldehyde. Okay, C double bond H. Okay, So this it is alcohol dehydrogenase is nothing but oxidation or reduction, whether which reaction it is carrying out. Okay, there are other types of enzyme nomenclature also which are used. So one of the these are the different types. So it could, first is trivial name. So what is this trivial name? It could be so this trivial name, it doesn't tell us about any source or function of the reaction. Okay, so it is just a random name which is allotted to the enzyme. For example, pepsin. Okay, so this pepsin enzyme, if I tell you that this pepsin enzyme, so you won't be able to guess what this pepsin enzyme is doing if you have no clue about this, the name pepsin or diastase, for example. We don't know what it is doing, okay, or where it is found. So we have no clue about the source. We have no clue about the source or function. Okay, so it's just a trivial name. Other example is chymotrypsin. So we don't know where it is, where the enzyme is found or what the reaction it is catalyzing. Okay, so they are just trivial names which are given to the enzyme, okay, during the time of discovery. And these trivial name, they doesn't give any information about what is the source of or what is the function of this enzymes. Okay, next we can also name the enzyme based upon substrate. So there are few enzymes like maltase. Okay, maltase. Okay, now how it is labeled as maltase? So it is substrate based, that is the maltose is the substrate. Okay, maltose is a carbohydrate, and it is broken down by maltase into glucose. Okay, it is broken down by maltase. So we are taking a substrate name, that is maltase. We are taking half of the substrate name malt. Okay, and we are adding the suffix ase, that is the suffix used for all the enzymes. Okay, whenever any name ends with ase, we know that it's an enzyme. So we are adding ase to the suffix by using the substrate name, that is maltose. Okay, so there are many enzymes, examples of enzymes which are labeled after the substrate. Okay, similarly based upon the source, okay, of enzyme. So source of enzyme as in where the enzyme is found out. For example, we have papain. Okay, this papain enzyme, it is found in papaya. Okay, this papain enzyme is found in papaya and it is capable of hydrolyzing protein. Okay, it hydrolyzes protein. Okay, so from papain came from papaya. Okay, so this tells us about the source of enzyme. Okay, so that's why the name papain, because it came from papaya. Okay, similarly there are systemic name based upon the reaction they catalyzed. Okay, so for example, if they are catalyzing some kinase reaction. Okay, what are the kinase reaction? That is transfer of phosphate groups. Okay, transfer of phosphate groups. Okay, so let's take an example of fructokinase. Okay, fructokinase. So again the substrate here is fructose. Okay, which is actually been used. Okay, fructose, which is then it is fructose is converted to fructose 1-phosphate. Okay, fructose is converted to fructose 1-phosphate by help of fructose 1-phosphate kinase. Okay, so so now it is telling, so the systemic name is telling us about the substrate, which is fructose, and it is also telling us about the what reaction it is catalyzing, that is kinase. It is catalyzing kinase reaction. So ATP is involved here. Okay, the ATP, from ATP the phosphate group will transfer on to the fructose. So this belongs to the transferase class of enzymes. Okay, and then we're left with ADP, right? We'll be left with ADP. So this fructose will get a phosphate group attached to it with help of the enzyme fructose 1 kinase enzyme or fructokinase, and then it will catalyze fructose 1 phosphate. So at C1 carbon, this enzyme, this phosphate group will get attached. Okay. So now we'll move on to the assignment questions from the previous years. Okay, so this might help you in solving the assignments from the for this year. Okay, so the term catalysis was introduced by, so this term catalysis was first introduced by Johannes Jacob Berzelius. Okay, he was a Swedish guy. Okay, he's a Swedish guy who Swedish scientist who saw this observation by Kirchhoff. Okay, what he observed, that there was some catalysis. So there was catalysis of starch. Okay, starch was converted to glucose with help of HCl. Okay, starch was converted to glucose with help of HCl, but what he observed, that the HCl is again getting, okay, HCl is not getting used up by the starch. And he observed there is no consumption of HCl. Okay, so here the catalyst was HCl. Okay, here the catalyst was HCl. So this was observation made by Kirchhoff. Okay, but the term catalysis was introduced by Johannes Jacob Berzelius. Okay, so the correct answer is this. Okay, so he coined the term catalysis. Okay, the technique that utilized by John Kendrew and his team to determine the first protein structure in 1958 was, so here the protein can be visualized by either electron microscopy, we can do it by NMR, X-ray crystallography, and mass spec. But in 1958, the first protein structure, it was X-ray crystallography was used. Okay, so X-ray crystallography is very powerful tool, and still we use X-ray crystallography, and it is very old also. Okay, we didn't have the other tools which we now have in order to determine the protein structure, but X-ray crystallography still now is one of the best tool to study different protein structures. Okay, so yes, the answer is C, X-ray crystallography. This was used. So it is very difficult to find out protein structure from NMR. Electron microscopy will still give us some sense of protein structure, but very detailed minute molecular structures can be found out by X-ray crystallography. Again, mass spec will give us about amino acid sequences. It will tell us about sequences. Similarly, NMR will also tell us about sequences, what are sequences present in the in the protein, but the overall 3D structure can be only found out by electron microscopy and X-ray crystallography. Okay. The drug that was produced from 9 enzyme cascade in 2020 was dash. So these are so whenever the drug ends with VIR, okay, these are antiviral drugs. Okay, and these VIR specifically are anti-HIV drugs. Okay, these are anti-HIV drugs. Okay, and the answer is A. Okay, the answer is A. So what is this enzyme cascade? So 9 enzyme cascade. So basically 9 enzyme cascade refers to different type of reaction. Here 9 different reactions. Okay, we have 9 different reactions occurring simultaneously one after the other, and we have 9 different enzyme used. Okay, 9 different enzyme used in order to catalyze this product, okay, in order to catalyze this product. So we have different enzyme used here, okay. We have different enzyme used in order to catalyze this reaction, and that is why this is known as 9 cascade enzyme reactions, okay. Okay, so these are the so these all reactions occur one after the other. That's why they are termed as cascade reactions, and these are the 9 enzyme cascade reaction. That is different enzymes. Okay, all different 9 types of enzymes are used to catalyze these reactions. Okay. Okay. Specific location on an enzyme where the substrate binds and the catalytic reaction occurs is A, active zone, B, reaction center, C, active site, and D, binding region. So the correct answer is C, active site. Okay, so active. So we have a protein. We have a 3D protein structure, and then we have a active site here. Okay. So the substrate will come and bind to this active site. Okay. So the correct answer is active site. Very simple. The specificity of enzyme towards substrates and product is due to A, temperature conditions, B, geometric complementarity, C, pH fluctuations, and D, allosteric regulation. So here they are asking specifically about the specificity of enzyme, that is how specific the substrate or how specific the enzyme will identify the substrate, okay, and that specificity will come from geometric complementarity. Okay, so what is geometric complementarity? Geometric complementarity is nothing but there is some so we studied that specificity could be because of either geometric, it could be because of electronic complementarity, right, electronic complementarity, and it could be also because of stereo specificity. Okay, it could be because of stereo specificity. So what is this geometric and electronic is that how is the active site pocket, how is the shape of the geometric shape of this active site pocket, and how it will fit the substrate in order to go different types of, in order to have a proper binding. Okay, so this geometric complementarity is very essential for the specificity. If the active site is too small or if active site is too big, there won't be any specificity. So example, if the active site is too small, okay, and if the substrate is too big, there won't be any specificity towards the enzyme and substrate. Okay, similarly, if the active site is too big, what will happen? If the active site is too big, what will happen? The substrate, many substrate at a time will come and bind, and there won't be any specificity as such observed. There won't be any contact, pretty much contact between the enzyme and substrate in order for the catalysis. Okay, and then there are other options like temperature conditions, pH fluctuation, and allosteric regulation. So again, temperature conditions is like we have, so how the rate of reaction proceed is, so we have temperature here on the x-axis, or it could be pH, both graph will look same. Temperature or pH on x-axis, and on y-axis we have rate of reaction. Okay, how fast the reaction is proceeding. So at lower temperature or lower pH, the rate will be very slow, whereas the optimum rate will be found at a particular range of temperature and pH. So temperature and pH basically are correlated to the rate of a reaction. Okay, if at a particular range of temperature and pH, we'll have optimal reaction conditions. Okay, there will be optimal reaction. Okay, and they will not correspond to the specificity of the enzyme. Similarly, allosteric regulation could be either positive or negative allosteric, and again it will tell us about how the reaction can be either positively regulated or negatively regulated. Okay, they will not tell us about how is the specificity or how is the binding of the substrate to the enzyme. Okay, next question is the role of nicotinamide adenine dinucleotide, that is NAD or NADP, in enzymatic reaction is what? So the options are transfer of acyl groups, carries high energy phosphate bonds, stabilizes DNA structures, and option D is act as carrier for electron and hydrogen ions. So as we know that NAD, so as you see from this reaction, so we have a NADH. Okay, this H is this. Okay, this H, this hydrogen here, what it does, it is attacking this carbon atom here. Okay, it is attacking the C double bond O. So the hydrogen is attacking the C double bond O, the hydrogen from the nicotinamide adenine molecule. It is attacking at this double bond between carbon and oxygen. Okay, with help of a zinc, with help of Zn2 cofactor. Okay, and what it is doing, it is, so this enzyme is alcohol dehydrogenase. So it is so what it is doing, we have CH2OH. Okay, we have a alcohol group, sorry, we have a aldehyde group here, alcohol dehydrogenase. So we have a aldehyde group, this is the aldehyde group, right? CHO is the aldehyde group. It is converting, sorry, the alcohol dehydrogenase, yeah. What it is doing, it is attacking to this, and to this will get, what will get, will get NAD plus, and will get ethanol, right? We'll get CH2, sorry, we'll get CH2OH. Okay, we'll get CH2OH. So this hydrogen group is hydrogen group will come and it will form so this is a reduction reaction. Okay, this is a reduction reaction. So we have from CHO, C double bond O, we are getting CH2. Okay, we are getting CH2OH. Okay. Okay, so this is, so the hydrogen group will be get transferred here. Okay. So yeah, so it act as a carrier for electron or hydrogen ions. Okay, carrier of electrons and hydrogen ions. Then coenzyme A, or it is known as CoA, function as carrier for what? So this is the structure of coenzyme A. So we have a adenosine 3,5-diphosphate. So this is the adenine group here. Okay, adenine nucleotide, and it is attached to a ribose sugar, and we have a phosphate group. Okay, and we have other two phosphate groups here. Okay, so this is adenosine 3,5-diphosphate, and then we have a phosphopantothenic acid. Okay, so this is the structure of coenzyme A. And finally we have mercaptoethylamine. Okay, here this is the structure of mercaptoethylamine. Okay, CONH. So this mercapto, because of this sulfate group here, and then we have a X. So now this X could be either hydrogen, okay, or in case of coenzyme A it is a hydrogen, or in case of acyl coenzyme A it could be a acyl group. Okay, COCH2. Okay, it could be either COCH2 or a hydrogen group. Okay, or hydrogen group. So what it is doing, it transfers this acyl group. Okay, it transfers this acyl group, okay, to the this coenzyme transfers the acyl groups. Okay, it has a X which can carry acyl groups. Okay, it can it is a carrier for acyl groups. Okay, so this coenzyme A, it is made up of this coenzyme A is made up of this vitamin pantothenic acid, and it will transfer acyl groups, okay, onto the substrates. So the correct answer is acyl groups. Then we need to match the coenzymes with the respective nutritional deficiency and from the options here. So we have coenzyme thymine pyrophosphate. Okay, so this thymine pyrophosphate, whenever we have deficiency of this enzyme, coenzyme thymine, so thymine is also one of the vitamin B, B2 I think, thymine pyrophosphate, and it is derived from vitamin B2, thymine pyrophosphate, and the deficiency leads to beriberi. Okay, it leads to nutritional deficiency beriberi. Similarly, NAD plus and NADP, nicotinamide adenine dinucleotide, can lead to pellagra. Okay, and the tetrahydrofolate is also a B12 folic acid vitamin. Okay, and the deficiency can lead to megaloblastic anemia. Okay, option B. And coenzyme B12. Okay, so now we have coenzyme B12, which is also involved in the different reactions, and it can also lead to pernicious anemia. Okay, so this both tetrahydrofolate and coenzyme B12, they are responsible for anemia. Okay, different types of anemia. So the correct option is D. Okay. The next question is, the information provided by EC number about enzyme is, so what information does the EC number provides? So options are its genetic sequence, its specific catalytic function, its structural conformation, or its cellular location. So EC number, as I said, it will tell us about which type of reaction they are catalyzing, right? Which type of reaction they are catalyzing. They will also tell us about something about substrate. What is the substrate? What are different electron or hydrogen or functional group donors, right? Different functional group donors are present. They will also tell us about what kind of reaction it is, what are the different type of reactions they are catalyzing. Okay, so basically they will tell us about specific catalytic function, right? They will tell us about specific catalytic function, like which is the type of reaction, whether it is oxidative or transferases, and they will give us information about where the reaction will occur, at what functional group of the substrate functional group, and also who will transfer that functional group. It could be either any coenzymes or different cofactors, and it will also tell us about substrate, what is the substrate. So it will tell us all the information. So EC number will tell us about specific catalytic function. Okay, so the next question is, the class of enzymes in EC system responsible for joining of two molecules using energy from ATP is A, EC6 ligases or synthetases, option B is EC3 hydrolases, option 2 is transferases, and option D is oxidative. So only one class of enzymes which leads to the formation of formation of or joining of two molecules, that is ligases or synthetases. Okay, so hydrolases again is breakdown, right? It will break down with help of water, right? That's why the name hydrolases, with help of water. Transferases, it will just transfer the functional group. There is no formation or breakdown. It will transfer functional group. Okay, and oxireductase, it will carry out oxidation and reduction reaction, right? Oxidation and reduction reactions. Then we have lysis also. What the lysis will do? It will again break down. Okay, it will also break down. It will break down, but it will break down without the help of water. And whenever there is joining, it's always ligases or synthetases. Okay, and whenever there is joining of two molecules, it will require energy, and that energy could be in form of ATP. An example of enzyme represented by EC 2.7.1.1 is the EC system. The EC system is dash. So the second type, so at first we have oxireductase, and the second one is transferase, right? The EC2 is transferases here. So they should be some transferases, that is transfer of different functional group, right? Transferases. So an alcohol dehydrogenases is oxidation reduction reaction, as we see in oxidation, so they belong to oxireductase. Kinases, yes, the correct answer is kinases, that is they will transfer the phosphate group. Okay, so we have hexokinase, we have fructokinase. Okay, so these are all kinases which will this belong to the EC class of transferases. Okay, they will transfer the functional group. Okay, and here the functional group is nothing but phosphate, right? The functional group is phosphate. It will transfer the phosphate from ATP to the different molecule like fructose or different sugars in hexokinase. Peptidase is again breakdown. Then it will be peptidases are it could be hydrolases, and ligases again formation. So they are different EC numbers. They have different EC numbers to them. Yeah, so here is the picture. So EC 2.7, so we have EC2 belongs to transferases classes, and we have subclasses. Here the 7 number, let's go to 7. It transferring the phosphorus containing group. Okay, the phosphorus containing group here are only kinase enzyme. Okay, so the correct answer is kinase. Okay. The biological process where cytochrome C oxidase, a type of oxireductase, participates. So we all know about electron transport chain. So we have different cytochrome oxidase. So here we have cytochrome oxidase C. Okay, and this cytochrome oxidase C, we have different sets of compartments with first 2, 3 cytochrome C4, and finally what they are doing, they are all carrying the electrons. So they get electrons from either NAD or NADH or FADH2. So they can get electrons from FADH2 and NADH2, and these electrons are transferred in form of, that's why the name electron transport chain. And the final electron acceptor is the hydrogen, right? The final electron acceptor is the oxygen, sorry. It will accept the electron and it will get water molecule, right? So cytochrome C oxidases are involved in electron transport chain. So they will accept electron, and they will undergo oxidation. So that's why, and similarly when they will transfer they will undergo reduction. So they belong to the oxidoreductase class. The type of name used for lysis when the reverse reaction is more important, or the only one demonstrated is. So don't get confused between synthase and synthetase. So lyses are the enzymes which are involved in breakdown of CC, CO and CN bond. Okay, So they involved in the breakdown of these bonds. Okay, so what are so in the in common names, the expressions like so here for example we have decarboxylase or aldolase. Okay, so here in these cases the reverse reaction is much more important. Okay, so that's why the name which use is synthase, okay, and not synthetase. Okay, so the reverse reaction is more important, is much more important, or only one side. So basically all the reactions are reversible in nature. Okay, but here uh, they only the reverse reaction are much important. Okay, the which are involved in decarboxylation or any dehydration of any reaction. Okay, elimination of uh, CO2 or aldehyde or water. So that's why the name synthesis. Okay, the name synthesis is used instead of synthetase. Okay, synthetase is like formation of any formation of synthetase refers to the formation of any molecule. Hydrolase is also breakdown, reductase is also breakdown. So when in lysis, when the reverse reaction is much more important, so we use the term synthase. The isomerase that catalyzes intramolecular rearrangement involving the transfer of functional group within the same molecule is racemase, epimerase, cis-trans isomerase, and mutase. So the EC class of isomerase has different subclasses. Okay, so it is a fifth class of isomerase, and it has different subclasses. So class 1 is racemase and epimerase. Okay, so this class, as the name suggests, it will give us different racemic mixtures of the chiral of the enantiomers. Okay, whereas the second class is the cis trans. It will carry out different isomerization of between cis and trans molecules. And the intramolecular isomerases. Okay, so the isomerases that catalyze the intramolecular rearrangement, as asked in this question, the intramolecular rearrangement. Okay, so this class is done by it is carried out by enzyme mutases. Okay, so this mutases, as you can see, they carry out intramolecular transferases reaction. Okay, or intramolecular rearrangement. So these are the mutases which belongs to the subclass 4. Okay, they carry out the intramolecular rearrangements. Okay, that is that functional group are transferred among the same molecule. Okay, the process that the catalyzed ligases, the processes that catalyzed by ligases in cellular function is breakdown of RNA, formation of phosphodiester bonds in DNA, synthesis of ATP, hydrolysis of protein. So ligases are involved in the formation of molecules which involved ATP use. Okay, so here we have T4 DNA ligase and we have E. coli DNA ligase. So what they are doing, they are carrying out this phosphodiester bond formation. Okay, so there is a phosphodiester, there is a bond between phosphate and the OH group. So there is a bond between phosphate and OH group, and this bond is known as phosphodiester bond. Okay, and it helps in joining of the two adjacent DNA molecules. Okay, so now after formation of phosphodiester bond, we have joining of these molecules together. Okay, so this was the final question. Any questions? Yeah, so any questions? Okay, if no more question, if you don't have any questions, then we can stop the meeting. Yeah, thank you so much for joining. Yes, anybody has any questions regarding today's assignments or any topics that you want to discuss? Okay, then I'll end the meeting. Thank you, everyone.